Okay, so this is live and recording is going. So let's, I'll just open the waiting room now. Let's see. Okay. All right, hello everyone. Let's just wait a minute while everyone joins and we'll get started. Okay, I think we have everything set to go. Welcome everyone to another FLUXNet Early Career Network seminar. Your host today is Teresa Jasbak from the Ohio State University. And we'll have a few of us in the background helping out field questions. If you have questions, please type them in the chat and we'll get to them. And we'll have a Q&A at the end as well. So Teresa, take it away.
Yeah, hello everyone. Good morning, good evening, depending where you are. So yeah, as Gilberto said, we're gonna have two speakers today, and we will start with Professor Gil Bohrer. Gil is a professor at Ohio State University in the Civil and Environmental and Geodetic Engineering department, and he's gonna present today about his work. About the tree-hydrodynamic revolution, like how stomata regulation moved from the soil to the tree. And yeah, good.
Hi, thank you, Teresa, for the introduction. And for this audience, I'm also the PI of the University of Michigan Biological Station Flux Cluster, though it's not in my university. I'm running the flux site there, and a bunch of Ohio wetland flux sites. So trees are hydraulic systems. The whole purpose of stems, roots, branches is all about moving water from the root up to the leaves. And the first one to realize it was Leonardo da Vinci. He was a very obsessive guy. He needed to draw a bush in the corner of the background of one of his pictures. And he got stuck on the question, how thick should the branches be after they branch? So if we know what the thickness of the stem is, once it's splitting, how wide should the splits be? And we know it because he wrote it in the Da Vinci Code, which is the actual kind of diary of Leonardo. And he solved it from a hydrodynamic point of view. So he said, okay, because the point of the branches is to move water, they need to be like irrigation canals. And here, if you split irrigation canal, the total width of the canal should be equal everywhere in the system. So he understood conservation of mass of water. And he assumed that it has to be the same thing for trees and bushes. And his drawing is beautifully wrong because irrigation canals are rectangular and on fixed depth. So yes, the width should add up to the same. But branches are circular, so we actually should conserve the hydrodynamic. If we keep Leonardo's argument, we should preserve the cross-sectional, the sum of the cross-sectional area of the branches along those arches and not the branch diameters. But in reality, is neither of those is somewhere in between. But still, he made the point that the purpose of the branches and the stem is to move water, and we should analyze the tree as a hydrodynamic mechanism, which was amazing. And it took a long time to get back to that view. So all Earth system models totally ignore this fine point. They represent trees as a bunch of leaf, basically either a cloud or a sheet of leaf hovering within the canopy air, which is either the boundary condition for the atmosphere or part of the first layer of the atmosphere. And they have stomata, which control the fluxes of water. And stomata are controlled by some average soil water availability. The only recognition of the hydrodynamic properties of trees is that there is some rooting depth, but the rooting depth only informs us which soil layers need to be incorporated in the single average number of soil moisture that ends up controlling stomata. I sound like I'm complaining, but it's not a terrible way. We need to simplify in a model anyhow, and current models of evapotranspiration are actually quite accurate and doing quite a decent job. But we can do better, and with specific phenomena that if we want to resolve, we need to do better, and I will show those in my talk. So what's special with what I call a tree-hydrodynamic model, and I kind of made up this term, it's straight up, but it's not a well-accepted term. Different people may call it something else, but the essence in what I call a hydrodynamic model is that the control of the stomata is not done by the soil water. The control of the stomata is done by the xylem water potential. Now, in order to even resolve the xylem water potential, a hydrodynamic model then must represent xylem as a conductive creature with some pool of water storage within the xylem. Further on, we can extend that xylem into the soil. Some models do it, some do not. And then the soil water act as a boundary condition to the root, and then water move up the root stem system, the xylem system, and then the water potential in the xylem is controlling stomata. Is that exactly what's happening in plants? No, it's more complicated. We cannot ever represent the full splitting of the branches. The real control of stomata is the water potential in the sheath of cell that surround the stomata guard cell, and there's a few layers of tissue to move from the leaf xylem to there, and even in the more complex model, they include multiple levels. These are multiple levels of stem xylem. So we're still not exactly taking the water all the way to the stomata as the real plant, but we are closer. We are closer by adding this term of xylem water potential and xylem conductivity and a dynamic term of xylem conductivity as a porous medium that changes with the amount of water in it. Great. So before I show you what models do, let's look at reality. Can we identify phenomena in reality where we need to even go through all this complication of what xylem water potential? So all the examples I show you are from my site in UMBS, very beautiful site. You're invited to come in the summer after COVID is over. It's a cool site. We measure a lot of things. We measure not only the regular flux measurements, we do very detailed leaf area measurements. We have a plot census where we measure annually the diameter of all the trees in the forest. That takes a lot of effort. We have stem water storage observations as of recently. We have subflow observations for almost 10 years now. And we have, these are essentially two sites which are on the same forest, but one site, UMD, is sitting over a disturbed, we disturbed the footprint of the UMD site. We did it on purpose. We girdled every early successional tree, kind of making an experiment of what would forest post-succession look like. We waited about 10 years. The trees that were supposed to die naturally with succession didn't die at all, so we hired a bunch of people, and this is my co-PI, Chris Goff. We didn't hire him, he just volunteered, and applied chainsaws to the trees to girdle them and kill them all. So we have one undisturbed plot, that's UMB, and one disturbed plot, which is UMD, where we push the forest into an extreme disturbance with a very rapid succession. And now about, how much is it? More than almost 15 years after we did that, the disturbance, you can see the dead trees kind of fallen, but the forest is as thick, as dense as the original forest, just with a different species composition. The cool thing about our subflow measurement, so we started in 2011, almost 10 years ago. This was my, at the time, undergrad research student, Ashley Mateni, that by now have matured to a professor in Texas. And I have my academic grandchild, Anna, that's doing those measurements right now. And we show that by measuring a lot of trees, and we have about 200 trees where we measure subflow, but this paper was done when we had only about 80, that's already enough. Again, we don't have an infinitely heterogeneous forest. We have only about 6, 7 species that we care about. So if we measure more than 5 to 10 trees of each species, sampling the medium ones and large ones, we can reconstruct the total subflow to match, more or less match, what we measure with eddy covariance. So eddy covariance measure total ET. And total ET is soil evaporation, evaporation from leaf surfaces, and the evaporation of trees. But you don't know what species does what. With the subflux, we are measuring only evapotranspiration. But because it's coming from individual trees, we can scale it to represent the species-specific transpiration. Now, if it was radically different, then a species-specific transpiration is somewhat off. But because they are more or less the same, and again, we are off by about 20%, and remember that the error of energy closure of a subflux system is around 10%, so I'm not so depressed about 20% off. So I think we have a good plot level representation of species-specific transpiration rates, and the analysis I will show are based on these. So what did we see? So here is the original plot, species-specific. Okay, that's a feature of, and here is the species-specific transpiration. No, I'm sorry, that doesn't work at all. Break that. So the biggest trees, aspen, they are the early successionals. They are the one we killed in the disturbance, so they take most of the evaporation. Here you see transpiration rate per meter square leaf. So this has nothing to do with how common that species in the forest. This is more related to how sun dominant the tree is and what the tree is doing. So aspens are transpiring the highest rate, then maples and oaks and pines are down here. In the disturbed plot, so same time, same year, just same species, just we remove all the aspens. So pine grew up like crazy, oak grew up by about 100%, and maple is depressed. And we started asking ourselves, why is that? Why are maples being depressed when clearly oak and pine rise up because aspen is not there to shade them? That makes a lot of sense. But why is pine depressed about it? Why is maple depressed about it? So we assume it has to be some hydraulic issue, and we compare the total ET from the sites. We bin them by soil moisture content, and we see that as soon as soil moisture content drops below 4%, which we identify as the onset of stress, we start seeing difference between the control site and the disturbed site. So the control site and the disturbed site are transpiring exactly the same when the soil is wet. When the soil is dry, the control site is transpiring more. So stress is affecting the disturbance site more than it is affecting the control site. So something in the new species composition, by removing aspen, the new species, they can't handle stresses as easily as the aspen could. Okay, probing deeper into that. This is northern Michigan. It's very, very wet, but it's also very sandy. So water drains from the soil very quickly. So it's about to have about 10 days of no rain that you get what we call a mini drought, where soil moisture drop far, here's soil moisture, far below 4%. And as soon as we hit that threshold, 4%, which you write about here, you see that, and this is subflux. So this is daily transpiration. This is half hourly, but each spike is a day. Red is maple, blue is oak. And you see that at the 4%, maple starts shutting down its stomata, whereas oak seem to not care much. Now the rain came, it rained at this day, 225. And the soil moisture went back even higher than it was before it all started. But oak is back as it ever was, whereas maple doesn't really recover. It takes a very long time, like all the way up to here, about 15 days, to get to the full stomata conductance, the full subflux rate that it had before that drought event. So again, indicating, what that is indicating is that soil moisture is not necessarily the right control for stomata conductance, because if it was soil moisture, we should see an immediate return after the rain. And the other, now focusing deeper on that, we see that as we go and dry, the peak hour of subflux is earlier and earlier during the day. So it's around 11:30 a.m. here, and it's around 9 a.m. here. And again, the soil doesn't dry much during the day. And if the soil dried enough during the first day to close stomata after 11 a.m., stomata should be totally closed every day after. And again, that again is not the case. So if we were to take stomata control directly from soil moisture to the leaf, we will have problems in that response in representing the daily and the intradaily dynamics of stomata of subflux within a drying sequence when the onset of water stress is happening. That phenomenon of making your daily peak subflux early and early in the day is creating an effect of hysteresis. So if you think of the daily cycle of VPD or PAR, VPD will peak around 2 p.m., PAR will peak around noon, or in Michigan, it's more around 1 p.m. So if we follow stomata conductance as a function of VPD, if that's a physical function, we expect stomata conductance to be the same when VPD is the same. Or if to be fair, when VPD and light are at the same combination. But if we look at stomata response to VPD and light before noon, it typically is higher than in the afternoon. So the afternoon stomata conductance for the same physical controls is lower than the before noon. Again, if we were to represent stomata conductance as a simple function of light, VPD, and soil moisture, it is extremely hard to generate that kind of hysteresis. And that hysteresis is common even when the soil is not stressing. And we think that the real reason for that hysteresis is that plants transpire in the morning from the xylem water. So there's water stored in the xylem, they're an easy source. Once all the easy water is gone, now plants need to move the water all the way up from the soil through the roots, and that takes time. So when they get to that point, transpiration is draining the leaves and the water potential increase. Some plants are very sensitive and will close stomata at that point. Now, we can see that soil moisture on average does affect stomata conductance. So this is the hour of day, and this is stomata conductance in wet day versus dry day. But in the morning, it's more or less the same. It's just more complicated dynamic issue than a simple linear control of stomata conductance. And here I calculated stomata conductance directly from this combination of the subflux and the meteorological forcing using Penman-Monteith equation, kind of fitting it. Now, if we look at that hysteresis, the question is, does this hysteresis really exist? Is that, now this is hysteresis of stomata conductance. So this is not subflux. This is basically incorporating all the physical drivers. So when we have everything that drives stomata conductance externally, which is light, VPD, and so on, the same, stomata conductance should be the same. And we see that at least in the early afternoon and the late morning, it is not the same. We have this significant area of hysteresis, which indicate to me that the reason for the hysteresis is hydrodynamic. And then we get, as we get to dry days, hysteresis gets more severe. Now, part of this hysteresis is the control of leaves. So if you are evaporating water from the xylem early in the morning, the xylem water potential will increase during the day because it's harder to bring water all the way through the root up the stem than to lose the water to the air if you're at the point of the leaf. So some trees are sensitive, and as soon as xylem water potential drops a little bit, they immediately close stomata. What that will create is an isohydric response, where you see that the midday stomata conductance is always constant. So it doesn't matter how dry the soil is, and the only reason to do that is basically close. This is this is midday, sorry, this is not midday stomata conductance. This is the leaf water potential. So the leaf water potential is constant. This is by definition what isohydric mean. And maples are known to be very isohydric. So as soil moisture decreases, the only way to keep your water potential constant is to basically close stomata. As soon as your water potential drops a little bit, you close stomata, you stop it from dropping any further. Oaks don't do that. If the soil is dry, oaks are letting it go and water potential in the xylem drops a lot. However, what we notice, there's a trade-off. So the same maples, which are very isohydric and sensitive, if we look at the pre-dawn, and pre-dawn is after the tree had the whole night to recharge its branches, so the leaves are full of water, as full as they'll ever get. If the soil is dry, maple have a problem, whereas if the soil is dry, oak doesn't have a problem. We studied further with radioactive isotope, and we discovered that oaks just have very deep root systems, and they essentially tap straight to the lake aquifer, which is very, very deep, whereas maple have shallow roots and have to live with the soil moisture. Let's skip that, go to modeling. So here's my original model fetch. It was a branching model, way too complicated, completely not a realistic way to use in a large models, but making very cool videos. So we see as the day cycles through, we start at 8 a.m., sun is up, transpiration goes, water leaves the leaf, the color marks the xylem water potential, and as transpiration starts in the morning, why are you not continuing now? I don't know. There. The long branches and the top of the tree get very stressed, and then in the afternoon, stomata close, the sun goes down, and roots recharge everything back. And we can apply a simplified version of that, that's the fetch 2, which has only a single beam for the stem, and we show that that can improve our estimates of evapotranspiration in a forest plot. There we compare to a subflow data set from New Jersey. Very good fit, slope close to 1. The model is very convincing. We're very happy with it. The cool things that you can do with the model is things that are very hard to do from observation. So now we can use the model as a surrogate to understand the hydraulic properties of the trees. So for example, we can fit, once we fit the xylem conductance properties and so on, and get a very realistic fit to both the total and the species-specific subflow, we can then conclude that the reason that we see those phenomena are because of the difference in the parameters between those trees. And we see that anisohydric trees will stress earlier in the day, whereas oaks, which are deep-rooted, will stress much less and keep the peak transpiration more or less in the midday. And we are working now on incorporating the same hydrodynamic principle in the E3SM model. That's by harnessing the amazing power of Gautam Bisht, and hopefully that will be published soon. And Margaret made me tell you, and I'm very proud to tell you, this is the year of water fluxes. Ameriflux is having the year of, last year was year of methane, this year is year of water fluxes. There are many ways where you can get involved in year of water fluxes activities, including special issues in journals, special sessions in conference and meetings. There's some dedicated proposal for that with the USDA ARM, all kinds of other stuff. Talk to Margaret, chat with the Ameriflux team about that. And I'm done here, and we're happy to chat about questions and stuff.
Thank you, Gil, for the interesting presentation. So we'll have time for 1 or 2 questions, like quick questions. You can write them in the chat. So I already saw a question by Ryan. How similar is soil moisture content between the control and disturbed sites?
It is very similar. So the control, the disturbed site has slightly higher sand content. So the soil moisture is slightly lower, but it's a difference of point something of a percent. So very, very small difference in soil moisture between them. What we see is because of the dynamic, because it's at the beginning, it was a little bit more open. So we had more soil evaporation, even though that we had less transpiration when the soil gets stressed. So yes, there are differences in the soil moisture, but these differences don't explain why the different species grow intermingled in the same site. So there's no way that the maple will get stressed in the same site that the oak will. The oak will transpire more than the control, and the maple transpire less than the control. And both control and disturbed oaks and maples each grow in the same, near each other. They experience the same soil moisture. Well, they don't, because oak has deep roots.
Okay.
Yes.
We have
So
another question.
Assaad, yes. Oak basically have very deep roots, so it's experiencing a different soil moisture than the maple. And the maple is very sensitive to the surface soil. The pine too.
Okay. Does anyone have like more questions? Yeah. Yeah. There's a. Okay. No, I have asked already. Okay. If you have like more questions, you can like write them in the chat, and then we can come back to them like at the end. And now we're gonna move to our second talk yeah, thank you Gil for the presentation, and we're gonna move now for second talk by Assaad Mrad. Assaad is a postdoc at UC Irvine and Wake Forest University, and he graduated like recently from Duke University, and he's gonna present about modeling the spread of gas bubbles inside plants and their effect on photosynthesis during drought. So yeah, Assaad, you can go.
Thank you for the introduction, Theresia. I'm going to share my screen. And can you see my slides?
Yes.
Okay, thank you everyone for joining me here today for my talk. It's called Plant Water Transport and Photosynthesis in Water-Limited Environments. Most of this work, all of this work was done for my PhD at Duke University. I graduated just last December. And over the course of this work, I had many collaborators whose names you can see in the middle of the screen. The focus of the presentation here is the mathematical representation of these processes and the challenges that this representation entails. The main challenge is how to succinctly represent these processes. The history of plant water transport starts with medical doctors. In the 17th century, we had medical doctors like William Harvey and Marcello Malpighi, who were the first to make comparisons between animals with pulsating hearts and other animals like snails and plants with no pumping mechanism whatsoever. And they also made the first discoveries of capillaries in plants too, using the microscope. A century later, Stephen Hales pioneered the cohesion theory in plant water, in which he hypothesized that cohesion between plant water molecules was important for the transpiration stream. And a century later, Heinrich Cotta identified sapwood as the main sap conductive pathway. But at this point, there was a debate between so-called vitalists and physicists, in which vitalists believed that live cells were necessary for the transport of water in plants. And it was only until Edward Strasburger in 1891 that this was settled. He exposed a maple plant to copper sulfate, which is a poison to that plant. And even after the plant died, he still saw transpiration, which indicated that dead cells were only responsible for water movement in plants. However, he did not speculate as to the driving force of transpiration inside plants. But a few years later, Dixon and Jolly, who were inspired by Strasburger's experiments and devised an experiment of their own in which they pressured a branch with its leaves in a pressure chamber up to 3 megapascal, at which point they still saw dew forming on the surface of the pressure chamber. This indicated that tension also was part of the picture here when it comes to plant water or sap. And so they pioneered the cohesion tension theory as the driving force for sap flow in plants. A few decades later, Gottlieb Haberlandt realized that sapwood is in fact an unsaturated porous medium with both air and water inside of it. And this is because water is under tension, and unless it is fully sealed, there will be a risk of air entering plants. So my presentation here follows this line of work. I will first, in the first two chapters of this presentation, I will be talking about sapwood and plant water transport under drought. And I will also finish this presentation by talking about how photosynthesis is affected by drought because of plant cavitation or drought-induced embolism. So sapwood, you can see sapwood in the middle here, in the middle figure, that's on the right, in fact. It's just what this shows is a microscopic image of branch sapwood. And what you see is multiple pores, which represent the conduits inside the sapwood. And these are the main pathways for water to go from the roots to the leaves. And you can see that a lot of these conduits are near each other. This is because water needs to move from one conduit to the next, and so on its way from the roots to the leaves. Because in fact, not one conduit goes all the way from the roots to the leaves. This is very rare that it happens. So you need a lot of them connected to each other to have a conductive pathway for the water. Here on the right, however, I'm showing one of the conduits air-filled or embolized. When that happens, this conduit doesn't allow water to move anymore. And so what then happens is that the water needs to find an alternative pathway around that embolized conduit on its way to the leaf. So the presence of air increases the hydraulic resistance of the branch or organ you're trying to measure. And in fact, when water is scarce in the soil, more air enters the branch because the tension of the water increases. A convenient way that scientists quantify a species' resistance to drought-induced embolism is to plot the percent loss of conductance of that branch or the plant against decreasing xylem water pressure. And here what you're seeing, and these are called vulnerability curves. And here what you're seeing are three vulnerability curves from three different species. There's a shrub from Intermountain, Wyoming. It's called the hoary leaf. And what you can take from this figure is that it is much better at resisting drought than these two other species, because it takes a much lower tension for it to lose a significant amount of conductance. And scientists usually use one specific point, the P50 point, that you see using the orange horizontal line to quantify a species' resistance to drought. But there's also the slope of the vulnerability curve that's important too. To determine what affects a species' resistance to drought, we look at sapwood. Sapwood consists of anatomical elements ranging many spatial scales. You have the pore diameters that I've showed you before. They're on the micrometer scale. Conduits also have lengths, and they're usually on the centimeter scale. But I've also said that conduits are connected, and water needs to be able to move from one conduit to the next. And the pores that form the contact walls between the conduits are at the nanometer scale. So there is such a big range of spatial scales which complexifies the problem we're looking at. The objective of this work is to go from what's on the left, the anatomy, to what's on the right, the different vulnerability curve. So this is an upscaling exercise. And to do this, I built a computational model on MATLAB to simulate water flow and gas bubble spread inside Sapwood. Water flow encounters two resistances inside Sapwood. As mentioned before, water moves along the conduits, and these are assumed circular tubes. And so they are, this type of flow is described using the Hagen-Poiseuille equation. And there's also the flow of water from conduit to the other, and that occurs through the pit membranes. There are little membranes, thin membranes that exist on the wall connecting the two conduits, and each of these membranes contains a lot of tiny pores, nanometer pores. And this type of flow is described by a Sampson flow equation. What about gas bubble spread? Let's say you have two conduits connected. One is embolized, or gas or air filled, and the other is functional and water filled. When there's an abundance of water, the pressure difference between the air and the water is negligible. And the pit membrane and the pores, through capillarity and using surface tension forces, are effective at blocking air from spreading. However, what happens when the pressure difference between air and water increases is that the pit membrane starts stretching up to a threshold pressure difference, where the stretch becomes too large and the surface tension forces become unable anymore to stop the gas bubble from spreading. At this point, we have air seeding, in which the air seeds a previously functional vessel. And that at that point, this new conduit or vessel becomes non-functional too. And by the way, angiosperms like maples and oaks, they're conduits, we call them vessels. Because I'm going to use them interchangeably here, because I'm studying angiosperms particularly here. And the model in the end looks like this. On the top right, specifically, I'm showing a two-dimensional version of the model where the conduits are shown as black vertical lines, and the contact walls are shown as horizontal red lines. Of course, contact walls are just conduits connected. There's no lines or anything in real life, but they're shown here just for visualization. On the left is a drawing of what an actual xylem network would look like, with one of the conduits embolized. And on the bottom, I'm showing what a pit membrane looks like. And there are thousands of these between every two connected conduit. And on each circular pit membrane, there are many tiny pores. And it's the leakiest of these pores, the largest, that allows bubble to spread when the threshold pressure is exceeded. So how does gas bubble spreads in the model? How does it happen? So what I'm showing here is a grid of xylem, a xylem network, a two-dimensional form of the model. As I said before, the black vertical lines are the functional conduits. The red horizontal lines are the functional contact walls. At a delta P of 0, there are random bubbles throughout the network in blue. As the pressure difference starts increasing, you can see delta P increasing on the top, the bubbles start expanding throughout the network. And as that happens, other vessels become isolated. And they're here drawn in magenta. What isolated means is that some of these vessels are air-filled, but they are unable to move the water anywhere else in the network because of an adjacent embolism. And so by plotting the hydraulic conductance of that xylem network, with every pressure difference, we can simulate the vulnerability curve of the species we're studying. This is how vulnerability curves are generated using the computational model. And we've used this model using anatomical data only for Rocky Mountain maple and other maple species. And we're able to reproduce the vulnerability curves. Here I'm showing the example of Rocky Mountain maple. In the middle figure, there's the measured vulnerability curve in black with the standard deviation. And in orange is the model vulnerability curve. And we were able to reproduce the vulnerability curve using only anatomical data. And this was promising, but there was clearly a missing piece, because when I tried extrapolating to other species, this relationship broke down, or the model broke down. So there was a missing piece. And the answer is the middle scale. Between anatomy and the branch or whole plant, there's the network scale. And this represents the patterns of connectivity between the conduits. Some examples of these network scale properties are shown in these 4 different cross sections I'm showing here from 4 different species. So these are branch cross sections. On the top left, we're seeing a species that has mostly solitary vessels. And the vessels, by the way, are the white circular areas. These are the conduits of water in that species. And you can see that they're mostly solitary. And other species like oaks, they're still solitary, but there's a new conductive cell type, the conductive tracheids, surrounding these vessels. You have species that have mostly radial arrangements of vessels, and others like elms that have mostly tangential arrangements of vessels. And also notice how oaks and elms are ring porous species, meaning that they have larger vessels in their early wood and tinier ones in the late wood. And so even if the anatomy was the same for all 4 species, the patterns of connectivity between the conduits will affect how a species responds to drought. To study that effect, we tap into a field of physics and math called graph theory. And this approach specifically works for bubble spread dynamics for reasons I'm gonna explain. But on the left is just a drawing of a xylem network with 4 conduits in this case, connected to each other through contact walls drawn in red. What we do is collapse each vessel onto one node. So vessel 1 becomes node 1, vessel 2 node 2. And if vessel 1 and vessel 2 are connected, we draw an edge between them, between the 2 nodes, as you can see from the red edge between node 1 and node 2. So the reason this only works for bubble spread is because we're collapsing the vessels. And that works because we assume, and there's evidence for it, that bubbles, as soon as air seeding occurs, they expand instantaneously and fill a whole vessel. Whereas water, in fact, needs to cross the whole length of the conduit. So it only works for bubbles here. And then we count for each node how many connections it has. For example, node number 3 has 3 connections to node 2, node 1, node 4, and therefore we say its connectivity is 3. What we're interested in is the average connectivity of the whole network. So we take a xylem simulation, or a real xylem, and we calculate the average connectivity of its vessels, and then we can ask questions like, how does the average connectivity of vessels or conduits affect a species' resistance to drought? And to do this, we start with a hypothesis that there's a trade-off when you increase connectivity, you increase redundancy to water flow, which is good, but we also might increase the speed of the embolism spread inside the plant, which is bad for resistance to drought. To explain this further, I'm highlighting 2 vessels here that aren't connected in the current state. However, had they been connected, you can imagine the water on the bottom vessel being able to escape the xylem network to the outlet much more easily than in the current network, because currently it needs to move 4 vessels to the left to escape. And so increasing average connectivity will decrease the resistance of that xylem network to water flow, reduce the resistance to water flow. But there's also the issue of redundancy. Notice how at a pressure difference of around 2 megapascal, that conduit becomes isolated, so colored in magenta. Had the 2 previously highlighted vessels been connected, it wouldn't have been, because the water would still have been able to escape to the right. So this is how increasing average connectivity, on average, increases the redundancy to water flow and increases the number of pathways of least resistance for water. However, how does spread speed for embolism or gas bubbles increase? Look at that embolism or embolized vessel. And notice how at a certain pressure difference, it spreads to the left. Had it been connected to the vessel on the right too, it would have spread faster. And because the spread of bubbles, like the spread of diseases, is a multiplicative process, it will have a large impact on a resistance of a species to drought. And so this is how the trade-off happens between redundancy and spread speed of the gas bubbles when connectivity increases. But embolism spread speed is closely related to the percolation threshold. Let me explain percolation very quickly here. Percolation theory applies to your morning coffee too, in fact. And an easy way to explain it is by looking at the figure on the left. You can see a fluid colored in red on the bottom and the top of the grid. And in the current form of the grid, the fluid cannot flow from the bottom to the top. This is because the pixels in blue, which are the connected pixels, are very low in density. And so the conductive pixels in blue don't form a conductive cluster. But what you can do is increase the density of these pixels, and you will have, and you notice that the fluid is better able to penetrate into the grid up to a certain threshold density of conductive pixels called the percolation threshold, at which point the fluid can flow from the bottom to the top. And so we're going to use this theory, but the fluid being gas, the gas bubbles, to evaluate the process of embolism spread speed in xylem. And to do this, I'm just showing here that I expanded the 2D model to a 3D model to correctly look at maples. Look at the bottom, a box-elder species with mostly radial conductive clusters of conduits. And at the top, there's the modeled version, which has the same patterns of conduit connectivity, mostly in radial arrangements. So as I started increasing the average connectivity of modeled maple, the percolation threshold decreases. What that means is that as the average connectivity of conduits increases, in fact, embolisms have a much easier time spreading into the network, which confirms the hypothesis of spread speed increasing with connectivity. But there's also what happens with redundancy. Here on the right, I'm showing on the abscissa average connectivity like before, and there's two scatter plots. There's the blue scatter plot, which represents spread speed. And as mentioned before, after above an average connectivity of 3, it blows up. What about redundancy and the orange scatter plot? It increases significantly and monotonically between a connectivity of 2.5 and 3, but then plateaus. And to get the overall effect of connectivity on a vulnerability curve, we divide the effect of spread speed by redundancy. And we see here that there's clearly an optimum of average connectivity between 2.7 and 3. That I also have, I encourage you to go to the paper I'm mentioning here on the left to see how connectivity actually affects maple vulnerability curves. I look at 7 maples, but for the interest of time, I'm gonna be moving forward here and discussing how what we've learned about sapwood and plant water use under drought affects photosynthesis. Because remember, water to go to the leaf and for transpiration needs to go from the soil to the root through the xylem of the root and the sapwood of the root and the sapwood of the stem and the branch and through the leaf to, in the end, transpire. And so the photosynthesis is sensitive to all of these in series. And the question is, how do stomata and photosynthesis respond to a lack of water in the soil? And the stomata, just to remind you, are tiny pores usually at the bottom of the leaves, and they can open or close based on the guard cells, on the trigger pressure of the guard cells. And when they open, they allow gas exchange. They allow carbon dioxide to enter the leaf, but they also allow water to leave. And that puts the leaf and the branch and the whole plant, in fact, under the risk of drying. So there's a trade-off here to opening stomata, because you do gain carbon dioxide for photosynthesis, but you also lose a lot of water. So how do soil root and xylem hydraulic supply limits shape the economics of the Gibbs gas exchange? That was the question we were trying to answer here. To do this, we tap into the calculus of variations. So this is a field that allows you to formulate an objective function to maximize, which here is called the Hamiltonian. So the Hamiltonian is just an objective function that you maximize over a period of time, as I'm going to explain. So this is the Hamiltonian. It depends on the stomatal conductance, GS, on time T, on soil moisture X, and what is called the Lagrange multiplier lambda. The Hamiltonian is dependent on the carbon gain with time, not carbon gain, but the CO2 gain from the atmosphere by the leaves. But it also depends on the hydrologic balance, which just says that there needs to be conservation of mass, conservation of water in the soil. So whatever comes in needs to go out one way or the other. And the role of the Lagrange multiplier is to make sure that the objective function abides by the constraint of hydrologic balance. I'm also quoting another paper here on the right that has an even more detailed description of the modeling. But what we want to do eventually is to take the integral of that objective function, the Hamiltonian, over a dry down period and maximize it. So the hypothesis here is that the plant maximizes carbon gain for a cost of water over a dry down period. But we can't only include the Hamiltonian because there needs to be a terminal gain term. The reason is because different species, even in the same environment, will have different water use strategies, and we need to take that into account. This is what the terminal gain term JT does. JT is dependent on the soil moisture at the end of dry down. And you can see here an additional plant level degree of freedom, big lambda. And what a high lambda does is that it prescribes a conservative water use strategy. But if you lower the value of that big lambda, it becomes an aggressive water use strategy. So here we now see how we can predict the trends in stomatal conductance while also taking into account water use strategy. In the paper, I was also interested in seeing how different plant level vulnerability curves affect isohydric to anisohydric responses to drought. Here you're seeing the three scenarios I've used. And this is slightly different from a vulnerability curve because the ordinate does not have the percent loss of conductance. It actually has the root to leaf conductance. So it's not a dimensionless value. It's a dimensional one. But they do represent the same thing. But in this case, this is of the whole plant, not just of one branch. One of the scenarios was a vulnerable plant, and the other two were resistant plants, but one with a steeper and the other more gradual vulnerability curve. And it turns out that differences in whole plant vulnerability to cavitation or embolism spread do propagate onto anisohydric and isohydric photosynthetic responses to drought. And I encourage you to go to that same paper to see the results I'm talking about. But to end my presentation, I wanted to show you the test for the model using a very nice experiment from a great paper I read in New Phytologist by Venturas and others in 2018, in which they subjected 16 aspen species to 87 days of drought. And on the left, I'm showing the comparison between data in black and the model in red. What I'm showing here is the stomatal conductance against the soil water potential. And you can see a very good agreement between the two. And this gave us confidence that the model is giving us the right thing. And this ends my presentation. I want to thank you all for listening to me. And I'll be very happy to listen to your questions too and answering them.
Yeah. Thank you, Assaad, for your presentation. Yeah, if you have questions, please type them in the chat. Yes, there's a question. Is there a preprint available for your review? new physiology paper?
Yes, there is actually. In fact, let me copy the link to the preprint on the chat so you can have access to it. Here we go.
Let me just jump. I have a quick question, but just to let everyone know, you can mute yourselves now and ask questions as well if you prefer to do that way. Typing in the chat also works. I have a quick question for Assaad. You mentioned briefly some field data that you used to do benchmarking. I'm wondering if there's any other types of field data that would be useful to you to do benchmarking against this model that you developed. This is very interesting. Thank you.
Yeah, definitely. I think the data set there, what was available was definitely temporal trend in soil moisture, of course, rooting depth, and also xylem characteristics. So being able to know the vulnerability curves. Because as Gil presented before, knowing only the soil water potential is really insufficient to translate onto stomatal conductance. We also had meteorological inputs like temperature, relative humidity, radiation, whether it's a cloudy day or not. And so getting these were very important for benchmarking.
Yeah, there's a question from Susanna. So I was wondering, the last model you showed has been contrasted with data from halophyte species like mangrove stress. It would be super interesting because they are under soil stress.
Yes, that's a very good question. Not yet, but what I've just shown is the response of photosynthesis to drought, which is a disturbance. Salt stress does that, and it also depends on whether your species can filter the salts well in the roots or not. And that will contribute to salt stress. The advantage of the calculus of variations method and the introduction of the Hamiltonian term, if you remember, I showed a Lagrange multiplier multiplied by a hydrologic balance term. You could in fact do the same, but instead of a hydraulic balance, you have a salt balance term that takes into account the salt stress. And by doing that, you're subjecting the leaf to whatever stress, like whatever the stress of salt is at the root level, but even at the leaf level, in case salt does actually go into the plant and stress the leaf. I haven't done it yet, but that's definitely an area I'm interested to go into.
Okay, yeah, again, you can unmute yourself anytime to ask questions. So there is a question for Gil. How would you go about doing a similar evaluation of sensitivity to drought in an ecosystem with much higher biodiversity?
We thought technically we can try to bundle up species. So collecting data is hard. So you'll always run into some limit of how many subflow sensors you can install and monitor. But in theory, that system doesn't have to work at the species specific level. So each tree is different. The point is, can we bundle trees up to groups of similarly behaving trees? So right now in my forest, all the trees of the same species and similar size group I put together. Though if you look at the subflux, there's quite a variation between trees based on their individual character and whimsies. But in theory, we can go to tropical forests, identify 3 or 4 different distinct hydrodynamically functional groups, and then try to sample trees within those and focus on the larger trees, which hold most of the transpiration.
Yeah, thank you, Gil. Assaad, do you have also questions? You mentioned that gas and water coexist in the xylem. network at the beginning of representation. Gartner 2014 et al. 2014 showed that gas inside wood has an important role of tree biomechanics. Do you know if there are studies relating this evidence with the amount of embolism a plant can resist?
In fact, no, I'm not aware of any studies that make direct connections between the hydraulic resistance of sapwood to drought and the biomechanics. But in terms of the ability to sustain air inside the sapwood, most of the studies I think at least the ones I'm aware of, relate wood density to both biomechanics and hydraulic vulnerability to cavitation, because wood density does help determine the two. The actual mechanism is, I don't think it's very clear at the moment, but I don't think there are explanations in terms of the presence of air. And I think this is a very novel topic. I don't think it's been done much before.
Yeah, you have another question, Assaad. Thank you for the nice and clear presentation. For the connection between vessels, is the pressure over time taken into account for the air to expand in vessel 3 to the connection point with vessel 4, for example?
Sorry, yes. The pressure is, of course, because you need a certain pressure difference between the air and the water for air seeding to occur. So if, let's say, vessel 3 has air, but vessel 4 does not, air does not just go instantaneously to vessel 4. That depends on the properties of the connection between vessel 3 and 4. For example, how big the pores are in connecting the two. In terms of time, once air seeding occurs because of a big enough pressure difference, we say that the air seeding occurs instantaneously. So we're actually taking pressure into account, but not time. I hope that answers your question.
Can I ask it this way, because it might be easier to understand?
Yes, sure.
So it's actually, so you have an embolism occurring at the top of vessel 3, for example, and it's more the time it takes to expand to the point where it connects to vessel 4. So the connection to vessel 4 is at the bottom, for example, and for it to get from the top of vessel 3 all the way down.
Yes, that's a very good question. Let's say the embolism starts at the top of vessel 3, but the assumption we make is that when a bubble exists, once a bubble starts existing in a stable manner in a vessel, it expands instantaneously to the whole vessel. And so once it starts existing at the top of a vessel, it instantaneously reaches the bottom. I know this is an assumption, but there is also biophysical calculations that have been made to calculate that, and the time it takes is very small.
Okay, thank you.
Okay, so it's already 10, so people, you're free to go and free to stay for more discussion. So there is a question to both of you, Gil and Assaad. How far do the plants travel down their vulnerability curves in the simulations, and how quickly can they return again?
So in my model, they return. Basically, stomata conductance is a function of xylem water potential, and xylem water potential is a function of water availability. So once they cavitate, conductivity goes down, so it's slightly harder to recharge. But as soon as they recharge, they know they forgot about it. So it's perfectly plastic, the cavitation curve. There's no hysteresis of cavitation, which I think is the weak part of the model. But there's many other issues to solve first. So let's get the model up and running and functioning. But a very elegant next topic will be to focus on the dynamics of recovery from stress. So I think the model plants travel too far down and recover too quickly.
I will also...
My model, Assaad model, I don't know.
Yeah, my model doesn't take it into account because I only model just one dry down period. So, but I would also add that in the xylem studies, the plant physiology community, I think there is a debate, a heated debate, in fact, whether hydraulic recovery occurs. So let's say if a vessel is embolized and has a gas bubble, if the plant is able to remove that gas bubble and put water instead and make it functional again, I think there's conflicting evidence. I think the more common way plants survive a dry down events is through the building of a new sapwood layer in the growing season.
So Daniel showed that there is damage also to the pit itself, and which further will justify building new tissue rather than hydraulically correcting the embolism, which should be much harder than we think.
Yeah, really great. It's a great answer, because I've been wondering about this. You know, so what happens is we see a drought, right? And we see that sort of transpiration goes down. But then, as you showed, Gil, when the water comes, there's another rainfall, then it takes maybe a few days in some species. In the others, it's almost immediate. Transpiration goes back up again. But I mean, if they have to build new vessels for that, that wouldn't really be possible, right? So I'm sort of wondering whether if we do this through the hydraulics, whether we are not getting the right results for the wrong reasons.
So in theory, what the hydrodynamic assumption does is put the stomata regulation to prevent cavitation. So if you calibrate your stomata correctly, your stomata will close before you dive too deep into the cavitation curve. And then, but the problem is then, according to that model, the plants recover too quickly because all they have to do is reopen stomata as soon as the xylem recharge. So there must be some hysteretic mechanism to force the stomata to remember that it just experienced some serious stress and not open so quickly. Because we see the trees recover within 4 days to a week, but not take the whole season. So,
okay, so this would actually not be caused by cavitation at all. It would just be preemptive stomata closure.
Correct.
That makes sense. Okay, yeah, thanks.
Probably something in between. Physiologically, it's probably something in between. It's some preemptive response which is slower than stomata opening and closing, some protein or something like that.
Gil, if you would like to expand on the question that you answered in the chat about the recommendation on using or revising the Penman-Monteith equation at forest and stand level, or is the model still valid?
So I like Penman-Monteith equation. So basically, it's flexible enough. So if you have real data, you can easily parameterize it to be surprisingly and incredibly accurate, which to me indicate that it's a very decent model. I've had a lot of success with, instead of putting the aerodynamic resistance using a Monod-Bukov term, using kind of a U-star-based aerodynamic resistance. It's from a book, which then is known, escape my brain right now because you're looking at me and you're stressing me out. But yeah, that's easy and common. So as long as you got the aerodynamic resistance right, you can easily calibrate the stomata conductance such that Penman-Monteith will be very reasonable.
Okay, so yeah, thank you so much, Gil and Assaad, for being part of the seminar. Thank you for the audience, for the great presentations and the great questions. And yeah, looking forward for our next seminar. Thank you.
Thank you.
Bye.